Neyi söylemek istediniz? Pardon, duymadım.
Sesinizin geldiğini onaylamak istedim hocam.
Ha, tamam, tamam. Başlayayım mı sunuma?
Süreniz geldiyse hocam.
Yes, dear guests and participants, welcome to this session. Now, this session, we have almost also 6 presentation again, and Professor Sama Awad Abdurrahim and Elizabeth Paitan are the session chairs. I think the session chairs are online. No. Okay. The first presentation is Aslı Elif Tanur Samancı. The title is In Vitro Antimicrobial Activities of Salon-B Products. Also, the second presentation is second presentation will be given from Aslı Elif Tanur Sabancı, and the title Investigation of Antioxidant Properties of Propolis Products from the World Market. You can give your both presentations, Dr. Aslı Elif. We couldn't hear your voice, Dr. Aslı Elif. Could you please open the microphone?
Do you hear my voice? Can you see my presentation?
Yes.
Sorry, I'm trying to share my presentation in vitro antimicrobial activities, but I cannot.
We are seeing your presentation in the screen, but please make full screen.
But what you see is the other one.
Okay. Yeah,
but
we are seeing the other one.
That's the problem.
No, that's not problem. If it is not problem for you, that's not problem for us.
Okay. Thank you very much. Today I will be talking about the antioxidant properties of propolis products from the world market. We tested propolis samples, propolis product samples from the world market in Istanbul Technical University Excellence Center of Bee Products. This center is specialized for testing of bee products like royal jelly, honey, pollen, and propolis. In this center, there is an expertise on beekeeping, agricultural engineering, R&D, food science, and technology. And there is a capacity to test nearly everything about bee products and other food products like chemical analysis, physical testing, analysis of residues, authenticity, and etc. I hope that everybody knows what propolis is. It's a resin-like bee product that bees collect from the trees. And as you can see on the picture, it looks like a stone when you collect it from the hive. It's a natural, very healthy, a superfood, a very good bee product. But there's a lot of adulteration about this product on the market. Normally, natural propolis, raw propolis, contains 50 to 60% resins, 40% beeswax, 10% essential oils, about 5% pollen, and other materials. There are a lot of impurities when you collect it from the hive. And as you can see, there's about 40% beeswax in raw propolis, which our body cannot synthesize. So our body cannot digest beeswax, and it needs extraction. There are different extraction methods in literature about propolis, but the best extraction methods described is done with ethanol, glycol, glycerol, so components like alcohols or alcohol derivatives. Extraction method of propolis affects its biological activity. So it is very, very important. It should be extracted in a manner that there should be no residues of beeswax in the resulting extract, and there should be all the active compounds in the resulting product. So the aim of the study was to determine the antioxidant activity of propolis products from the market. So we collected 50 raw propolis samples from Turkey, and we tested them for their wax content, total phenolics, flavonoids, and antioxidants. And we collected 130 propolis samples from the market, world market, in the form of extracts like drops, sprays, syrups, and other liquids. These were generally sold in stores, pharmacies, and online shopping websites from 19 different countries. We tested them for their dry matters, total phenolics, flavonoids, antioxidants, and phenolic profiles. As you can see from the picture, we collected 50 raw propolis samples from different regions of Turkey. Most of the samples were from Marmara region. We also collected from Middle Anatolia, East Anatolia, Southeast Anatolia, Aegean region, Black Sea region, and also Mediterranean region. And here are the results for wax content, total phenolics, flavonoids, and antioxidants. As you can see, the highest antioxidant capacity is from Marmara region, and the lowest from southeastern Anatolia by means of number of according to the number of samples. And we also tested them for their flavonoids and phenolics, and the highest concentration of wax content is also from southeastern Anatolia. We also tested 130 samples from the world market, and you can see the countries here: Belgium, Brazil, Croatia, UNA, Estonia, France, Germany, Italy, Japan, Korea, Latvia, New Zealand, Romania, Russia, San Marino, Slovenia, Spain, Turkey, and USA. Most of the samples are from Turkey, 54 samples, and 18 samples from Korea, and the rest of the samples are from different countries. We collected all these samples from the world market between 2016 to 2019. We compared the analysis results of these samples from the world market with the Anatolian propolis we collected from Turkey, and we prepared as water-soluble extract 10% and ethanol extract 20%. And you can see the minimum concentration that we look for an antioxidant capacity of a propolis extract containing 10% pure propolis is 150. And for a 20% extract, we are expecting to have over 200 milligram Trolox per milliliter sample antioxidant capacity. And we are also expecting for fumaric acid, rutin, apigenin, camphorol, krisin, galangin, kafeic acid, ferulic acid, cinnamic acid, pinosembrin, kafeic acid, phenylethyester, quercetin, artepilin C, and genistein. Here are the results of the samples. We hide the brands here. You can see the content of the product, the dry matter, result of total phenolics, flavonoids, and antioxidants. And as you can see, many samples even doesn't contain any antioxidants, phenolics, and flavonoids. Here we mentioned in red, and most of the samples were not labeled according to the real propolis content. You can see here, as I'm going on the results of the samples, there are many results which have great problems for labeling and the content of the product. Here we have 4 samples which shows the expected amounts according to the propolis concentration. And if we calculate all the results totally and we compare the results with the Anatolian propolis, the results of the samples compared to the standard extracts containing 10% and 20% propolis that we prepared in our lab, you can see that the total average concentration of the samples from the market is 8 times less than the expected amounts. Again, total flavonoid content is 11 times less than the expected amounts, and the total phenolic content is 9 times less than the expected amounts. So there is a real standardization problem for propolis products in the Turkish market. 20 samples showed no antioxidant capacity. 28 samples had very few antioxidant capacity. 2 samples showed the expected concentration. Korea had a better situation, but nearly the same problem is again on the market. There is a problem of standardization and mislabeling. As you can see, we tested nearly all the countries have the same problem. Spain, US, UNA had a greater problem. Nearly none of the samples we tested showed antioxidant capacity and had a real concentration of propolis in the sample. USA and Japan nearly had the same problems. We have a normal sample from USA. Slovenia, again, the same standardization problem. And Germany also has the same problem. Belgium and Brazil also shows the same problem. And if we also look at the overall results from the world market, we can see that if you compare again with the standard Anatolian propolis, the results of the samples from the world market, we can see that total phenolic content is 4 times less than the expected amounts. Total flavonoid is 8 times less than the expected amount, and total antioxidant capacity is 3 times less than the expected amounts. So between 76 samples we tested, 29 samples showed no activity, 33 samples had very few antioxidant activity, less than the expected amount. 5 samples showed a better antioxidant activity, over 100. 9 samples showed the expected activity. Here I put a chromatogram showing the big difference from different samples. Here is a 15% propolis containing extract from Turkish market, and you can see another sample on the right containing 70% propolis extract. And you can see that in the chromatogram there is nearly nothing. It's on the label, it says it's 70% propolis, but when you look at the results of the analysis, there is nearly nothing in it. There is nearly no propolis. And a 15% propolis containing product shows the chromatogram on the left. So between the 20 samples showing antioxidant capacity over 100 milligram Trolox per milliliter, we tested their phenolic profiles by chromatography. And 13 samples didn't show any phenolics coming from propolis. So this proved that some samples on the market has an antioxidant capacity, but not coming from propolis. Maybe it's from vitamin C or something added in it, but it's not from propolis. And only 7 samples showed the expected concentrations and the expected amount of propolis mentioned on the label. So you can see the results of the chromatography here. As a result, 96% of all the samples were mislabeled about the propolis content of the product. 65 of the samples did not specify the amount of propolis on the label. The antioxidant capacity of 38 of the samples were below 10 milligram per milliliter, indicating that only trace amount of propolis or no propolis was present in the sample. Majority of the samples contained preservatives and additives that were not indicated on the label, misleading the consumer. It's obvious that a product which is regarded as a natural healing source by many people should be standardized on the market. Thus, consumers should not be misinformed about the content of the product, and the standard must be established to regulate the content of products and take unfair competition under control on the market. Thank you for your attention, and if you have any questions, I will be happy to answer your questions.
I think no questions, Dr. Elif. Maybe you can continue with the second presentation.
Okay. Sorry, I will try to open it now. Can you see the presentation? Sorry, can you see the presentation?
Not yet.
Okay.
Elif hocam kapatıp tekrar açmayı.
Evet, öyle yapıyorum şimdi. Onu deniyorum.
Şu anda sizin ekranı görebiliyoruz hocam.
Ha, ekranı görüyor musunuz?
Ekranınızı görebiliyoruz, evet.
Tamam, bir saniye. O zaman şuradan tekrar açmayı. Şimdi görebiliyor musunuz?
Evet, evet hocam. Bir de tam ekran yaparsanız.
Hı.
Tamam.
Tamam, görebiliyorsunuz.
Tamamdır hocam, buyurun.
Hello again. I will be describing the in vitro antimicrobial activities of Salon-B products in this presentation. We run the testing. It's a collaboration of 3 centers. We run the testing in BNU Propolis R&D Center, Karadeniz Technical University, and Recep Tayyip Erdoğan University. So the results that I will present here is coming from a collaborative project that we run all together. Here, our company, BEO and BNU, we have 2 brands. BNU is for our international market, BEO is for national market. is an award-winning company offering innovative value-added propolis-based food and food supplements to consumers seeking for healthy and natural products. And our facilities are registered for BRC, ISO 9000, ISO 22000, GMP, IFS, and also we have organic certification, halal and kosher certification too. In our research center, we are running all this testing for bee products in a 6,000 square meter area. Our plant is based on 6,000 square meter area. We have food scientists, chemists, biologists, beekeeping experts, and food technicians working in our R&D center. And we are running different projects with 20 different universities. We are working with contracted beekeeping. So all the products that we tested in the study are produced by contracted beekeeping and extracted with our technology in our plant, patented technology, and turned into different formulations. So I will be talking about all this, all the results of these different formulations. To collect the samples, we give equipment to our beekeepers. We tested their locations and we trained them for the production of propolis and other bee products. And then we extracted and tested them in our laboratory for their biological activity and food safety issues. Turkey is a second country after China with its number of beehives. We have 8 million beehives in Turkey, and there is 81,000 beekeepers working for production of bee products in Turkey. Turkey is the largest producer of pine honey in the world and one of the largest producers of honey also. With its rich range of flora and wide range of honey, Turkey is a very important biodiverse area for endemic plants. So we have very good quality bee products like honey, propolis, royal jelly, and pollen coming out of this natural environment. In this study, we investigated the antimicrobial properties of propolis extracts, drops, sprays, tablets, and mixtures with raw honey on 16 different microorganisms, gram-positive, gram-negative, fungi and yeast, and acid-alcohol-resistant bacteria. We also run a second study for comparing the antibacterial activities of honey samples from Anatolia, from Turkey, with honey samples from New Zealand, Manuka. So we tested astragalus, pine, chestnut, polyfloral honey from Hakkari, and we tested also Manuka cow factor 16, Manuka active 15, Manuka MGO 400, and Manuka MGO 550 on 12 different microorganisms. Again, gram-positive, gram-negative, fungi and yeast. Here are the microorganisms and diseases that they are related to. These are most pathogen bacteria causing different health problems in humans. For example, Staph aureus for causing upper respiratory tract infections, Enterococcus fecalis causing gastrointestinal diseases, Bacillus cereus causing foodborne vomiting, nausea, and diarrhea, Streptococcus mutans, dental caries, Streptococcus agalatia, meningitis, Lactobacillus aedephilus for dental caries, oral and dental problems, Lactobacillus caseus, dental caries again, Mycobacterium stigmatis representing bacterial species causing tuberculosis, Candida albicans and Candida tropicalis causing fungus, and Helicobacter pylori causing gastritis, reflux, and ulcer in humans. So these are very important pathogen bacteria. So we tested propolis as drops, throat spray, nasal spray, tablet, syrup mixed with honey containing 1, 2, and 10% propolis. And in the other study, we tested again astragalus, pine, chestnut, polyfloral, honey from Hakkari, and manuka, different types of manuka. And we tested them by agar well diffusion method and micro dilution method. Here are the samples that we tested for different organisms, for different samples for different organisms. Drops, agar well diffusion method, you can see here 30% and 15% drops, propolis drops. We tested them against ampicillin and tetracycline. And you can see that especially 30% drop, as much as the concentration of propolis in the drop, in the extract, the stronger the antibacterial activity against both gram-negative and gram-positive bacteria. So these two extracts, the water-soluble extract and the ethanolic-soluble extract, both have gram-negative and gram-positive bacteria antibacterial activity. And we tested them also against streptomycin for Mycobacterium stigmatis, and against fluconazole for yeast antifungal activity. And you can see that again, both extracts have nearly same effect on Mycobacterium stigmatis and yeast, and both show antifungal activity. Nasal spray containing 6% of propolis shows also strong gram-negative and gram-positive antibacterial activity against tetracycline and ampicillin again. And they also have both antifungal and anti-candida activity against yeast and fungus. Propolis mixture with pine honey. Here we have 10% of propolis concentration. Shows strong activity, antibacterial activity against gram-negative and gram-positive bacteria. And also shows antifungal activity and anti-yeast activity. Mixtures with royal jelly, propolis, and raw honey containing 1% and 2% propolis shows also strong gram-negative and gram-positive antibacterial activity against ampicillin again, and also antifungal activity too. Syrups, tablets, and nasal spray, they all have strong gram-negative, strong antibacterial activity against gram-negative bacteria, and strong antibacterial activity against gram-positive bacteria, against ampicillin, streptomycin, again here, and also shows antifungal activity against fluconazole. Both formulations also have the same activity. And propolis pine honey and propolis chestnut honey mixtures, they also show strong antibacterial activity against ampicillin, streptomycin, and fluconazole again, against gram-negative, gram-positive bacteria and yeast. The other mixtures also have the same effect. And what about manuka samples? Manuka samples are well known for their antibacterial activities worldwide. And when we compared honey from Hakkari region, chestnut honey, pine honey, and astragalus honey produced in Turkey, Anatolia, we saw that even all these different 4 types of honey has more stronger antibacterial activity compared to manuka honey samples in the market. They all have also very strong activity against Bacillus cereus and Staphylococcus aureus, Bacillus subtilis and Klebsiella pneumoniae, Aspergillus niger, and Candida albicans. As a conclusion, the products in the form of drops inhibited all tested gram-negative and gram-positive bacteria species at different zone diameters. A significant antimicrobial activity has been observed, especially against Helicobacter pylori, Streptococcus aureus, Bacillus cereus, and Mycobacterium stigmatis. Throat spray form showed antimicrobial activity against 12 of the tested bacteria and yeast species. Measured inhibition zone diameters varied between 6 to 45 millimeters. The propolis raw honey mixture and propolis raw chestnut honey mixtures, which contains 10% propolis, showed antimicrobial activity against 12 different bacteria and yeast species. In propolis raw honey mixtures, the zone diameters ranged between 6 to 30 millimeters, and raw honey mixtures containing propolis and royal jelly were also found most effective on Helicobacter pylori and Mycobacterium stigmatis. As a conclusion, tablet showed antimicrobial activity on all tested microorganisms except for Pseudomonas aeruginosa, highly effective on gram-positive bacteria ranging from 12 to 26 millimeter inhibition zones, and the most sensitive one is Streptococcus mutans, causing oral health problems. And nasal spray showed high activity on gram-positive bacteria. Syrup showed antipseudomonal activity too. Pine honey and chestnut honeys are determined as the most strong effective honey types against Streptococcus aureus, and the antimicrobial activity of the chestnut honey against Escherichia coli, Strophacoccus frondi, and Bacillus cereus was higher than other honey types, and polyfloral honey from Hakkari showed strong antibacterial activity against two different bacterial species and Candida albicans. So thank you for your attention. And if you have any questions, I will be more than happy to answer.
Dear Dr. Aslı Elif, thanks for your nice presentations.
Thank you.
Yes, about bee products and propolis products, that was your presentations were very nice. Is there any question to Dr. Aslı Elif? I think there is no question. Thank you again. And in this session, now we are passing the third presenting author, Emrah Şirin, Dr. Emrah Şirin. He will present us about the significance of seed coat morphology in some Isatis, Brassicasa taxa.
Yes, I am here. Can I start? Can you hear me?
Yes, Dr. Emrah Şirin, we are hearing you, and now we are. Your presentation is on slide.
Okay.
Thank you. Yes.
My name is firstly, dear chairs and participants, welcome to my presentation. My name is Emrah Şirin. I am attending from Selçuk University. The title of my presentation is Significance of Seed Coat Morphology in Some Isatis taxa. Turkey is one of the richest countries in the world in terms of Brassicaceae species with 571 species. At the first volume of the Flora of Turkey, the number of Brassicaceae genus and species was indicated to be 86 and 464 respectively. These numbers increased to 88 genus and 526 species in the 10th volume, and to 91 genus and 555 species in the 11th volume. In the second supplement of Flora of Turkey, the total number of the species increased to 538 with the addition of one genus, 28 species 15 subspecies, and 2 varieties. Isatis includes about 79 species, and the genus is distributed primarily in the northern hemisphere, especially in the Eurasian region, where nearly 90% of its species grow. Isatis species sometimes show a morphologically highly polymorphic pattern. Seed surfaces, color, and size are valuable characteristics for distinguishing taxa in Brassicaceae. Therefore, the main objective of this study is to evaluate the taxonomic significance of the seed cotton patterns as seen by SEM in some taxa of the Isatis. The study material comprises samples of ripe seeds from 4 taxa belonging to Isatis collected from natural habitats in Turkey between 2015 and 2016. The samples were stored at Konya Selçuk University Faculty of Science Herbarium. The locations and collector registrations numbers of the taxa studied are provided in Table 1. The seed micromorphology characteristics are provided in Table 2. 20 seeds from each taxon were examined in our study. Samples examined in our analysis using scanning electron microscopy were first passed through a series of 20, 80, 96, and 100% alcohol for 20 minutes each. The surfaces were then observed and photographed with the Zeiss Evo LC10 scanning electron microscope at 30x, 1000x, and 2000x magnification in high vacuum mode. The seed micromorphology terminology used was according to Stern, Kaul et al., and Zeng et al. You can see localities of the studied taxa: Isatis arenaria from Konya, Isatis candoleana from Bayburt, Isatis cappadocica subspecies macrocarpa from Konya, Isatis erzurumica from Erzurum, and Isatis floribunda from Aksaray. The micrographs from the seeds of the 4 taxa studied are shown in Figure 1, and the macro and micromorphological properties of the seeds are provided in Table 2. Isatis arenaria, seed brown, narrowly elliptic, unwinged, glabrous, and the seed coat pattern is regularly reticulate. Isatis candoleana, seed dark brown to black, narrowly ovate, unwinged, glabrous, and the seed coat pattern is irregularly reticulate. Isatis cappadocica subspecies macrocarpa, seed dark brown to black, oblong, unwinged, glabrous, and the seed coat pattern is irregularly reticulate. Isatis erzurumica, seed brown, lanceolate, unwinged, glabrous, and the seed coat pattern is irregularly reticulate. And Isatis floribunda, seed dark brown to black, narrowly elliptic, unwinged, glabrous, and the seed coat pattern is rugose. Table 2, micro and morphological features of studied taxa. Figure 1, SEM micrographs of seeds of studied taxa Isatis arenaria from A to C, Isatis candoleana from D to F, Isatis cappadocica subspecies macrocarpa from G to I, Isatis erzurumica from G to L, and Isatis floribunda from M to O. SEM studies show that seeds, fruit, and leaf surface model characteristics are useful for describing different families and genera. Although Kara İsmailoğlu defined the seed shapes of Isatis arenaria and Isatis erzurumica as ellipticus, angus, and ovatus, respectively, it is more convenient to describe Isatis arenaria to be narrowly elliptic and Isatis erzurumica to be lanceolate according to Stern's system. These characteristics cannot be considered distinctive because the seeds of all taxa studied were wingless. Some other species within the same family that have a wingless seed structural Nasturtium officinale, Corypha islandica, Brassica napus, and Iberis linifolia. Isatis capodocica subspecies Macrocarpa has relatively larger seeds, while Isatis candoleana has relatively smaller seeds than those of other Isatis species. Kara İsmailoğlu reported the seed coat pattern of Isatis arenaria to be rugose and Isatis erzurumica to be smooth. However, it is more appropriate to interpret Isatis arenaria as regularly reticulate due to its regular whip-like structure on the seed surface, and Isatis erzurumica is irregularly reticulate due to the irregular whip-like structure on the seed surface. Color characteristics cannot be considered distinctive because the color of the seeds from all studied taxa brown or dark brown to black. Bona has observed a similar seed color in Lepidium species that are not related to those of Isatis. The seed surface of all taxa studied was glabrous. Similarly, Gampanach et al. Have reported a glabrous structure in Erysimum species that are not related to those of Isatis. The studied characteristics were not distinctive for Isatis in genus level, but the shape and size of the seeds and that coat patterns can be used as distinctive characteristics among the studied taxa. In future, seed morphology studies about the rest of the Isatis species will contribute to the interpretation of relationships of the taxa. The specimens were collected during the field trips for a project supported by a grant from Scientific Investigation Project Coordinator of Selçuk University. Thank you for listening to the presentation.
Thank you.
Yes, thanks for your nice presentation, Dr.
You are
Emrah Şive.
welcome.
Thank you. Is there any question? Okay. Another presentation is we're giving from Dr. Samah Awad Abdurrahim.
Yes, I'm fine.
Yes. The title is Evaluation of Antibacterial and Wound Healing Activities of the Combination of Funiculum vulgare, Cinnamon verum, and Schizirium aromaticum essential oils in diabetic septic wounds. Yes, please, Dr. Samahawat. We are ready to listen to you.
Okay, is it clear, please?
Yeah, please make full screen. Dr. Samawat, can you make
Yeah.
the full screen your presentation?
Sorry.
At the bottom of the file, you can find the full screen and you can press the full screen maybe.
My presentation is not clear.
Yeah, it is clear now, but it is not the full screen. We see the slide by slide.
Is it?
Yeah, that's okay. Yeah, perfect. Thank you.
Thank you. Good afternoon
Okay.
for all. Thanks for you for allowing me this opportunity to share my information with MESMAP-6 symposium. And I want to talk about the evaluation of antibacterial and wound healing activities of 3 essential oil combinations in diabetic septic wound. Of course, the wound infection in diabetic patient is a serious problem, usually affect the diabetic patient and causes lower extremity amputations. In addition to this, infection impairs wound healing also in the diabetic patients, as well as it is a major cause of hospital admission. This impairment usually due to the initial barrier of healing that are found in the diabetic patients. Because blood glucose level is high, causes cell wall to become rigid, impairing blood flow also in the critically small vessels at the wound surface. And also the process of phagocytosis is impaired here in the patient with the diabetes. So the diabetic infection takes longer time to heal because the delay of macrophage introduction and diminished leukocyte migration, which causes prolonged inflammatory phase in the wall, in the wounds, and all the wound healing cascade. This is an increasing cause of lower limb amputations, contributing to about 38% of amputations. So prompt research into the identification of new biocides with a broad activity are important because polymicrobial nature of diabetic foot infection usually increasing the resistance of these antimicrobial agents to the conventional chemicals. And the problem of resistance among diabetic patients is generalized. WHO in 2014 declared that the antimicrobial resistance problem is there. And also the Center of Disease Control and Prevention, which is an American center, declared that if we are not onslaught this revolution of resistance, we must return to the post-pre and the antibiotic era. So that we want to establish well indication for combination of antimicrobial therapy, and the combination usually is used for empirical treatment of life-threatening infections. This is in clinical settings. Also in the treatment of polymicrobial infections, prevention of the emergence of bacterial resistance, and also in the synergism. Usually synergism or combination chemotherapy has proved useful in the treatment of cancer, actually, and also in cardiovascular diseases. Here we use it in the bacterial infection. It's also previously used for viral infection. In addition, many biological processes are known to require the simultaneous action of 2 or more than 1 interacting agents to produce an stimulus effect. The synergistic effect from the association of antimicrobial agents, it is a new choice of the treatment because this effect enables to use respective antibiotic when no longer effective treatment by the self-therapeutic agent. So synergy is also taken to mean an attenuation of indesirable side effects. This is another key of herbalist theory that being that the toxicity of plant extract is less than using the single active constituent or single isolated constituent. Why we chose essential oil for this study? Because essential oils are complex in nature, and this make them not easily to be resisted, not looks like the single ingredient. So it can kill the microorganism or stop its growth from multiple target sites and multiple mechanisms of action. Also, it is lipophilic, which allows the topical application of it on the skin is to easily penetrate the subcutaneous lipid tissue of the skin and enter the blood circulation without affecting the normal flora that found in our gut. Also, it had antibiofilm formation. Syzygium aromaticum essential oils have proved that it has anti-quorum sensing phenomenon, and the quorum sensing actually increase the activity of bacteria to form the biofilms and then resist the antimicrobial agents. And also, as I said now, it has low toxicity when it compares with the single ingredient of the isolated antibiotics. So the objectives of this study is to assess the in vitro antibacterial activity of the essential oil combinations against standard control bacteria, against the standard control bacteria, and also against multiple drug-resistant aerobic bacteria associated with diabetic septic wound, and also to assess it in vivo, to assess the in vivo antibacterial activity, because the susceptibility test measures the antimicrobial activity of the antibiotic in the laboratory conditions only. That is to say, in vitro, not in the patients, in vivo. It cannot be assured, therefore, this an antimicrobial which is active in vitro will be active in vivo also, because there are many contributing factors for the drug pharmacokinetics, pharmacodynamics inside our body. So that the formulation of these essential oils in a way that the body can benefit from it in vivo, it is very important. We use here the albino rats, Western albino rats. Okay. It looks like a problem in the internet because the slide cannot be moved. Excuse me.
Yes, doctor. What's the problem?
I can't move the slide.
Maybe you can stop sharing and share again. Now I stop sharing. Please share your screen again.
Okay. No. Please stop sharing again.
Yes, now we are. That's okay. Your presentation is on our screen.
But I can't move the slide. Okay. So I want to speak about the methodology. This study was conducted in Khartoum, in Faculty of Medical Laboratory Sciences, and also in the Faculty of Pharmacy in the International University of Africa. And the ethical approach was obtained for human and animal subjects. For human subjects, what all procedures and techniques were in accordance with the national guidelines in the research conducted in human and animal subjects. And the protocol for human subject was approved from Sudan Federal Minister of Health, and for animal subject from the Institutional Animal Ethics Committee of the International University of Africa. And the plant material were purchased from the local supermarket, then authenticated in the Sudanese Medicinal and Aromatic Plant and Traditional Medicine Research Institute, via voucher specimen were deposited. Then the essential oil were extracted by hydrodistillation, and yield percentage was calculated. Other the minimum inhibitory concentration of these essential oils were determined in order to determine quantitative antibacterial activity, not qualitative, such as disk diffusion assays. And this in accordance with the Clinical Laboratory Standard Institute guidelines. The checkerboard test was used to assess the interaction, the synergistic interaction between the essential oil blends, and this required fractional inhibitory concentration calculation and fraction inhibitory concentration index, which is equal to minimum inhibitory concentration excuse me. The fractional inhibitory concentration is equal for a single blunt constituent is equal to the MIC of the essential oil 1 divided by in combination divided by the essential oil alone, and the FIC index equal the sum of FIC of A and FIC of B. I have told now about how we calculate the FIC index of each medicinal plant. And the in vitro screening, for just screening, we use the disk diffusion method. Here we use two types of bacteria, the standard control bacteria, American type culture collection for quality control purposes, and 76 clinical isolates of multiple drug resistant bacteria in order to see the activity of this plant extract in the clinical setting in vitro. So the isolates were belonging to the key indicators of bacteria that belonging to the resistant bacteria, methicillin resistant staphylococci and Escherichia coli, including 4 strains of extended spectrum beta-lactamase producing Escherichia coli and Pseudomonas aeruginosa as a representative organism for gram-negative organisms. Of course, Staphylococcus aureus is representative for gram-positive. All of them were collected from Khartoum, from Soba University Hospital. In order to perform the in vitro activity, we use adult in vivo activity, we use adult Western rats weighing not more than 375. And also the blunt extracts were formulated as ointments for mixture of 406,000 of polyethylene glycol polymer. This for formulation of ointment. Percent from each plant. This means that any plant extract was 0.3 just, and this is very low concentration. And the diabetes in albino rats was induced by alloxan monohydrate. Any rat showing blood glucose level more than 200.
Excuse me, Dr. Samawat. Please make conclusion your results and conclusion. Okay. Because we have time limits. Okay.
Okay. Our results here, this is the preliminary screening of this diffusion assay. The results varied from moderately active, such as here in the folliculum vulgare essential oil, and semi-complete zone of inhibition in Salmonella verum. And this is also for screening. MICs and MBCs, minimum inhibitory concentrations, was also promising because phenyl oil alone 0.78 against staphylococci, ranging from 0.781 to 25 in Pseudomonas aeruginosa. Here, the susceptibility of standard bacteria to essential oils in blends, FIC indices, all of them are less than 0.5, and this means synergism, because FIC index, if it calculated more than 0.5, this is called additive interaction. If it is more than 2 or equal to 2, this is called antagonism. All the bacteria are susceptible for essential oil in blends better than the one essential oils, and these are the picture of clinical isolates, multiple drug resistant clinical isolates. But as you see in this table, the p-value is not significant in the differences between gram-positive and gram-negative bacteria. Here, this diagram showed the contraction percentage of ointment in the excision wound. As you see in this diagram, the highest one is the herbal ointment. The herbal ointment showed 72% of wound contraction, followed by Fusiderm, and lastly, the treatment of the group. I want to say now, but the time not permits me, the groups of rats were divided into 3-1. This is the control group, which is left without treatment, contaminated just by bacteria. This is all of them contaminated by bacteria, but this left without treatment. Fusiderm group, which is a standard drug use, is used for quality control purposes and to compare the wound contraction percentage between essential oil and the standard drug use. This diagram here shows the wound diameter in the excision wound. As you see here, the diameter of control group is the more larger, 0.361. And between the standard drug and ointment, there is no significant difference between the herbal remedy and Fusiderm. This is the representative pictures of excision wounds in albino rat, in Western albino rat, and in the day 4. This is the viable bacterial count in the pus. The bacteria which was contaminated the wound initially was 10 raised to power 8 colony forming unit per ml. Here, there is a diminishing level of bacteria in the ointment into 10 raised to power 4 when compared to the control, which is 10 raised to power 5. These are histopathological pictures profiled by hematoxylin and using stain for the lesions induced in A2. Here we find in the ointment group good epithelialization, IEC referred to epithelialization. I see there is a scanty number of inflammatory cells, and this means that no bacteria is found here. The new blood vessels are numerous also. Compared with the Fusiderm group, there is new vessels, new vascularization, but not looks like this type of ointment. Here there is a numerous number of inflammatory cells, no blood vessels in the control group, so that we subjected these slides to form a Mason trichromostain in order to say which is a specialist stain for collagen formation. Here there is scanty number of collagen formation in the herbal ointment when it compares with Fusiderm group, which is a standard drug use. There is this blue coloration indicates collagen formation, but not intensively like the herbal group, so that in the day 11, this wound was completely healed. This is keratin layer indicate complete restoration to normal skin. So that this essential oil has in vitro antimicrobial activity and in vivo antibacterial and wound healing activity, so that we want to isolate the active constituent of this compound. I want to acknowledge my partners in this work and express my sincere gratitude to MESMAP organizing committee. Thank you for allowing me the time. Thank you.
Okay, thank you, Dr. Samahavat, for your nice presentation and sharing us your valuable research. Is there any question? Okay, thank you again. Now, our last two presentation will giving Dr. Nadire Pelin Bahadırlı. The first presentation title is Determination of Yield and Quality Characteristics of Different Coriandrum sativum populations in Eastern Mediterranean Conditions. And the last one, Inactivation of Klebsiella pneumonia and Staphylococcus aureus pathogens inoculated to rice by Lourdes nobilis and Tempera civicata essential oil. Dr. Pelin, you can summarize your both of the research because in 15 minutes.
In 15 minutes. Okay, I will be very fast.
If it's possible for you, okay.
Okay. First of all, thank you very much for the participants and also organization committee for making this Congress very fruitful, even with these difficult circumstances. My first presentation will be about coriander population and their cultivation in Eastern Mediterranean conditions. Coriandrum sativum is an annual culinary herb from Apiaceae family. As you know, Apiaceae is an important family with very economically important plant species such as carrot, cumin, and fennel. Coriandrum, also known as Chinese parsley, dania, cilantro, and also in Turkish it's known as kishnish, ashotu, and kuzbara. Here on the map you could see with the green sides where it is native to. These countries are Afghanistan, Iran, Syria, Pakistan, Palestine, and Turkey is one of them. And the purple ones are the introduced countries. So you can say that it's cultivated around the world. And in Turkey, there are two coriander species. One is Coriandrum sativum, and the other one is Coriandrum tordillium. Both of the species are not endemic, but Coriandrum sativum is the main cultivated species. Here you could see also in Turkey's map where they are distributed. In the Hatay region, where our experiments were done, there are only Coriandrum sativum species. All of the coriander parts, leaves and seeds are used in many industries, in food industries, in pharmaceutical industries, and also perfumery. Because this coriander has usage, because of its biological properties such as antioxidant, anti-helminthic, anti-inflammatory, anti-mutagenic, and etc. In Turkey, there are only 2 registered varieties exist. Before, in the past, there was also 4 more, but now only 2, Gürbüz and Arslan. Russia, Bulgaria, Mexico, US, Argentina, China, Romania, Italy, Japan, Hungary, Poland, Czech, Morocco, and India are the main cultivated countries. In Turkey, mostly cultivation is done for seed production, but around Erzurum province, there is a collection from nature for fresh herb and dry herb consume. Fresh herb essential oil from our previous studies results was 1-decanol, cyclodecanol, dodecanol, and decanol. The odor from fresh herb comes from decanol and trans to 3-decanol. Fresh leaves also contain vitamin A and vitamin C. The purpose of this study was to see the cultivation possibility of the coriander for fresh herb production in the Eastern Mediterranean conditions. We choose from many seeds, only 10 seeds. For their size and colors, we show a variation. Here you could see our description of populations and their seed size. We have only 3 populations which has a small seed size. Also in this picture, you could see all of our population seeds. Our experiment was carried out in Hatay Mustafa Kemal University Agricultural Research Center, which is very close to civilian board. Experiments were conducted in randomized complete block design with 3 replications. The experimental size was 7.2, and the space was, growing space was 0.3 meter. And for one, we used 2.5 kilogram per decar. Seeds were directly sowed into the field in 18th of November, and harvestings were done before stem elongation period on March. And in harvesting was in a different period because of their growing time, but in all of them was on March. Here you could see their harvesting times. And before sowing the seeds, we applied 4 kilogram phosphorus per decar and 4 kilogram nitrogen per decar. And plant height, number of branch per plant, fresh leaf rates, fresh herb yield, fresh herb essential oil rates, dry herb yields, and fresh herb essential oil yield was investigated. Here are some view from our field. As we look through our results, for plant heat, 4 populations had higher results from the other populations. The higher result was in population 9, 40.60, and number of branches were ranged between 3.40 to 5.03, and the highest populations was 6 and 5. Fresh leaf rate was only high in one population, in population 1, and it was found 44.88. And for per decade, fresh herb yield was very high in many populations, population 2, 4, and 7, 8, 9. Fresh herb essential oil rate was for coriander, it was normal, but for essential oil, for other essential oil containing plants, it was a bit low. It was the highest one was 0.03%. And in the dry herb, we couldn't determine any essential oil. Dry herb yield was high in population 2 and population 7. And population 2, it was 279.93. And fresh herb essential oil yield was high in population 2, which was 0.50 per decade in liter. So here are some other results for coriander. So in some studies, they continue for a seed production, but we harvested for fresh herb. That's why maybe our plant height was a little bit lower than the other results. For example, Kan and İpek found between 40.8 and 58.5, and Gümüşçü and friends found 49.7 to 68.7. Also, number of branches were found similar to our results, but also other researchers obtained more branches, such as in results from Kaya, Kan and İpek, Tunç-Türk and Tunç-Türk, and Qatar and Tunç-Türk. Fresh herb yields were ranged between 845.6 and 906.5 kilogram per decade in another study. Dry herb yield were found between 200.1 and 8, 220.5, and fresh herb and dry herb yields were higher from this result, higher in our results when we compare from this study. Mostly essential oil studies focused on seeds of coriander because it contains more essential oil. But leaf essential oil from another studies were found as 0.04, 0.1, 0.03. Only in one study from Buyan, they observed higher results when comparing with our results. This could be because of ecology, different soil types, and cultivation time, harvesting time, because secondary metabolites can be very, very easily because of ecology and other conditions. Population 2 from Altınözü, Hatay in our city, 4, 7, 8, and 9. 4 was from Burdur, 789 was commercial samples, found higher fresh herb yields, and only one essential oil yield was higher from Altınözü Hatay. But they had remarkable results for us for future studies. The results show that Hatay is suitable for cultivation of coriander for fresh herb consume. In Hatay region, the coriander seeds are only cultivated for seed production. There is no cultivation for fresh herb. Essential oil content found lower than in some studies, and as I explained, because it could be ecology, variety, soil type. There are so many effects that could change the secondary metabolites. And thank you for listening. I tried to be very fast. I hope it was clear.
Thank you for your presentation. That was clear, very nice. Thank you.
You're welcome. If any questions, I can.
Maybe we can take your second presentation, because actually the time is over for this session.
Okay.
So could you please just summarize your your research?
The second presentation will be about inactivation of Klebsiella pneumonia and Staphylococcus aureus pathogens inoculated to rice by Laurus nobilis and Timbra spicata essential oil. Medicinal and aromatic plants have been used for centuries for many purposes, including flowering, medicine, nutrition, pharmaceutical, cosmetics,
Excuse me, Dr. Pelin. Can you just summarize your results, please? Okay. Yes, we are sorry that, but time is over, okay?
Okay. Should I say the aim?
Yeah, please.
Okay. The aim was to see antimicrobial activity of Laurus nobilis and Timbra spicata essential oils on Klebsiella pneumonia and Staphylococcus aureus, as well as the logarithmic reduction of these microorganisms by these essential oils after the inoculation of the microbes to the rice. So I skipped this part. Okay. Here are the results for Laurus nobilis essential oils. As you see on the graphs, eucalyptol is the main content, which had 54.15 rates. And the other main component was alpha-terpinyl acetate, 16.28. For Timbra spicata essential oil, main component was carvacrol with the amount of 60.65%. And the other main component was gamma terpinen with 23.03 rate. So when we look through the antimicrobial activity of these essential oils, we see Timbra, for their MIC results, we see Timbra spicata essential oil had a really high results. And also, if we look through the, again, for the IZ results, Timbra spicata essential oil also had high results. In this figure, you could see inhibition zones and their effects on Klebsiella pneumonia and Staphylococcus aureus. And here is the Timbra spicata essential oil and the Laurus nobilis in both microbes. for log reduction, both of the essential oils had effects, but Timbra spicata had very high results with 150 microliter essential oil when applied on it. So Laurus nobilis essential oil and Timbra spicata essential oil components in many studies are the same. Eucalyptol are the main content for Laurus, and for Timbra spicata, carvacrol is the main component, but their range are varies in different studies. There are different results for Laurus nobilis against Klebsiella and Staphylococcus. In our study, it had lower effect, and this could be due to interactions of essential oil containing herbs or the microorganisms that studying may have gained resistance to these compounds. In contrast to that, Timbra spicata was highly effective against Klebsiella and Staphylococcus. Many studies in the literature are similar to ours. It seems possible that this activity was a result of high carvacrol and gamma-terpenen content in Timbra spicata. These are the results for Laurus nobilis essential oil main components, and these are the Timbra spicata main components. A significant difference was observed between Laurus nobilis and Timbra spicata essential oil in terms of their antimicrobial activity. It's observed that Timbra spicata had higher high effects on both of the diffusion and microdilution test. The reduction in microorganisms attached to rice was greater in the study with Timbra spicata. In this case, we could say Timbra spicata are more effective against bacteria that can penetrate on rice. And thank you for your attention.
Yes, thank you, Dr. Pelin, for your nice presentations. Is there any question? Okay, now we are completed this session, and I think we cannot give coffee break, and we will begin the last session now. Now I will share the program for the last session. For the last session, the session chairs are Şantru Karmendana and Gülsüm Yaldız are the session chairs. And first presenter author is Professor Marina Sipinu from Romania. If the session chairs and presenting authors are ready, we can begin this session.
Hello, Sevgi. It's very nice to see you. Can you hear me? I must apologize on behalf of Dana Chandru. She is busy with lectures. We have, because of COVID, lots of problems and crowding hours. And if you agree, I would like to chair instead of her this session. Is it a problem, or you want to nominate somebody else?
No, it is not a problem. If you have time to organize session, it is honor, pleasure for us. Thank you, Professor.
Thank you very much. Thank you. Is my colleague, my co-chair here present? Is the co-chair present so we can start?
Yes, we can start. The first presentation is yours.
Okay, so just allow me to start sharing my screen.
Okay, Professor.
One moment. Find my presentation. We are a little bit early, so I was not prepared with it on the screen. It will take 1 second to open it. And just to let you know, it is a presentation which is financed from the Horizon 2020 project we have. I'm not sure what I've done wrong. Okay, so I'm back and I'll try to. Can you see my screen now?
Okay.
Yes, Professor.
Yes.
We are. Yes. Please make full screen.
Yes, yes, in a
Yeah.
minute.
Okay. Thank you.
Okay. It was at its end. So the presentation is, sorry for that, called in vitro age-dependent cell-mediated response to antimicrobial plant extracts in free-range Mangalitsa pigs. It's the group of researchers that works in the infectious diseases department, part of project Horizon 2020, as I mentioned it before. And the concern of this project is to make sure that welfare and health of swine and birds, chickens in free range farms, on low input farms, is acquired. So there is recently an increase of the numbers of such farms, and in these farms where animals are free range, are in these conditions with access to outdoors, there is a significant microbial pressure against these animals, and therefore it's very important to preserve their health by natural means and decrease as much as possible morbidity and mortality of the livestock. In fact, the animals are in between two possible situations: good welfare and healthy immune system, or inadequate welfare, biased immune response. So our aim is to maintain them as much as possible in good health and good welfare. In between these, the factors which are influencing the good health and good immunity are housing, accommodation. As I mentioned, these animals are free range, so they have access to the outside, having the possibility to encounter more bacteria as normal. Then their feeding, what they eat, is very important as well. And this is also a way for us to provide them nutrients which have antibacterial and immune stimulating roles coming from plants. And then caretaking is also a very important issue where the administration, the management of the farm has to take care. The epidemiological pressure, as I said, is much higher. And due to this and lack of preventive means, you know probably that on any kind of extensive or organic farm, normal treatments, classical treatments, vaccinations are not really allowed to be used. And in fact, we lean on the native potential of the immune system to protect the host against diseases. Excessive therapies such as antibiotics administered excessively would impede on the health and welfare of the animals. Fortunately, this is not the case, and here plants provide another resource because, as we heard today and yesterday, many of them have antibiotic properties. So what we decided to try and do in this research is to elaborate strategies to improve health and welfare by experiments on sows and piglets conducted on low input outdoor farms in my part of Romania, which is northwestern and central Romania. We would like to achieve a combined antimicrobial and immune stimulating effect in all age categories of animals raised on such farms with access to pasture. Luckily, some of these plants we took into account are also available on these pastures. either naturally or administered as pelleted feed supplements with these medicinal plant extracts. And we were considering Cucurbita pepo, Coriandrum sativum, Allium sativum, and Calendula officinalis. I just listed here some of the compounds which are active in these plants because there are very many, and I'm sure that there are specialists who know them well because they work a lot on the chemistry of these compounds and isolating different patentive compounds from these plants. But I'm not going to go into detail on this chemical composition. It's just a list to see that some plants we chose share some compounds which are known for their antimicrobial effects. Our mission was in this experiment to look at their immune stimulating potential. So what we've done, we chose one age group from each category, 10 animals per group, suckling, weaned, and adult animals. We sampled blood from these animals, which we cultivated in vitro in 96 well plates in the presence of mitogens, known mitogens, such as phytohemagglutinin, and also adding to these cultures plant extracts using mainly alcoholic plant extracts, except coriandrum, where we use the essential oil. Again, these products were listed in the German pharmacopoeia and are commercial products. We did not prepare them. After incubating the plates for 48 hours at 37 degrees, we again measured the growth of the cells by quantifying the glucose residue spectrophotometrically, and we calculated the stimulation indices based on this glucose consumption that occurred during 48 hours. Then we compared the results using students' t-test for statistical significance of the results. These experiments were carried out, as you see, in vitro. This is the first stage. As I said earlier in Salon-A, our aim is first to, as much as we can, characterize the biological effects of these extracts in vitro and then use them, if possible, once well defined, in vivo. So these are some of our results. I indicated here in this graph the effects of medicinal plants we used in comparison with the control culture and the mitogen we use, the standard mitogen, phytohemagglutinin, and then the solvent control, which is alcohol. As I mentioned, we used mainly alcoholic extracts. And from this graph, you can see that the Calendula officinalis extract acted best in this age category, above the level of the control, untreated control, and alcohol control, while it was somewhat below the level of phytohemagglutinin, which is a very potent immune enhancer. The difference between the lowest, which was coriander, immune stimulating compound, and calendula, was of about 6%. In weaned piglets, older category, the difference was just 4.5%. And you see that most of the extracts acted inhibiting when compared to the alcohol. And at the same level, if we talk about garlic with the control, a little bit above the control. Nevertheless, this was below the level of alcohol. So we considered in this case that the garlic extract acted inhibiting compared to the solvent control. The best immune stimulating extract was that of the pumpkin, in this case, giving us some idea of what future extracts we could use in vivo, because this is food which pigs like very much, and it has very many nutritious facts besides being immune stimulating, as you see in the graph. Looking at the scale at the indices, percentages in adult animals, we see that most of the extracts acted very similar. Nevertheless, because of this range, which is much broader here in lesser space, we can see that the difference in source between the inhibiting compound and the highest stimulating effect, which we saw in calendula and cucurbita or pumpkin, was of 10%. So there were high differences between these extracts in adult animals. If we look at the statistical significance of the results, so we can see if we compare the piglets with the older categories, weaned piglets, all the differences are statistically significant for all the plant extracts, as opposed to those between the intermediate category of older animals but still young and adults, where there was significance just for coriander and garlic and pumpkin. What we concluded from this study, which is at its beginning and we still have further experiments to do, is that the plant extracts used for their known biological activities in this experiment impacted based on age of the animals and the plant family, proving some of them immune stimulating capacities which differ depending on the age of the animals. Calendula and cucurbita were most efficient in very young piglets, cucurbita in weaned piglets, and in sows, none of them looked very encouraging. And this is the group of very young PhD students accompanied with by our lab assistant, who worked a lot on this project, and he still have a lot of work to do. And of course, this was funded, as I said about it before, from the European Union's Horizon 2020 research project number 186172. And I thank you very much for your attention. If there are any questions, maybe I could answer.
I think no question, Professor Marina.
No questions. So I think then the second paper in the program is also our paper, and I would like to pass the word to my younger colleague from the group you've seen before, if it's possible, for her to present this second paper.
Yes, it's possible.
Okay, just give us 1 minute to share the presentation. Just one moment. Technology is sometimes very complicated in these COVID times.
If Professor Marina, if you need some break when beginning your second presentation, we can pass another author.
No, I just want to put it on the screen because it's right here. And I'll pass the word to my, can you see my screen? It's going to be full screen in a minute. Okay, is it okay?
That's okay.
Okay, thank you. So I'll pass the word to my colleague, Diana Ola.
Hello, can you hear me?
Yes, we can hear you.
Hello, I'm very happy to participate in your symposium. Thank you very much for having me. And please allow me to continue in the same trend that you've been used to from our department, our discipline, the trend of studying plant extracts. So here are some results of the study named In Vitro Adaptive Cellular Response to Alcoholic Plant Extracts in Brucella ovis infected rams. Brucella ovis was firstly reported in New Zealand in 1950 53, and it is an agent of alarming reproductive disorders. It produces the infectious orchioepididymitis in rams and high economic impacts in numerous sheep breeding countries, as it does in Romania. Brucellosis is a declarable disease in sheep, and it is included on the list of declarable diseases of the World Health Organization of animals. Failures to eradicate this disease worldwide, as in Romania, may be due to the lack of specificity and sensibility of diagnosis or surveillance tests, which are applied for maintaining the control of this disease, and also to the measures that are not properly taken. So the emergence of worldwide antibiotic resistance is an acute problem for the last decades, more so for the last years, even if it is not a new topic of discussion, because antibiotic resistance is ancient. It is a means of resistance in the environment of bacteria, a resistance to other antibiotic-producing bacteria, but the desire to discover alternative methods of treating bacterial pathogens or enhancing the ability of the immune system to fight disease has intensified over the last years more so. This study sought to determine to what extent the adaptive immune protection, which is mediated by mononucleosides like lymphocytes in Brucella ovis infection, can be influenced by extracts obtained from plants which animals can naturally graze on pastures in Romania. We carried our investigation on sheep raised in herds belonging to private breeders. Herds are raised on pastures in the northwest of Romania. They are mainly used for milk production. Usually the female sheep are maintained for production around 5 to 7 years, but the rams only 5 years because their productivity decreases and they are eliminated from the collective. Blood was sampled on heparin for the 3 groups of rams with positive, doubtful, and negative complement fixation tests, which you see here on the right. This is the confirmation test. The preliminary testing is done by Rose Bengal test. So this is the protocol that we apply here in Romania. An in vitro blast transformation test was carried out. We used the polyclonal mitogens, the classical mitogens, which are known to produce the transformation of blasts, phytohemagglutinin, concanavalin, alcohol, and alcoholic extracts of plants belonging to various families of plants, like Laminaceae, Ecvisetaceae, and you can see Taraxum officinale, Achillea millefolium, Salvia officinalis, Arctium lapa, Ecvisetum palustre, Avena sativa, and Hypericum perforatum. The samples were distributed in 96 wells plates. Afterwards, the extracts and the mitogens were added, and the plates were incubated for 63 hours at 37 degrees. Afterwards, the stimulation indices were calculated using the residual glucoses measurement. As you can see here in the graphics, the dynamics of the blastization indices for the 3 groups, for negative, for doubtful, for positive groups. In the control groups, there is a high variability between individuals, but overall, the positive group showed to be more receptive to the immunostimulating, to the lack of immunostimulation in the whole culture, in the whole blood cultures. Here you can see the results for the dandelion extract treated cultures, and the positive group showed the higher dynamics, the higher blastization indices compared to the negative group. The other way around happened to the group which was treated with yarrow extract. So overall, the negative group treated with Artium Lapa showed higher blastization indices. In SAGE, the results were pretty close, but higher blastization indices again in the negative group. The other way around was for burdock, where negative group, the RAMs which gave negative results to complement fixation tests showed higher blastization indices. For field horsetail, the positive group reacted better. As for the oat extract treated cultures, the positive group showed higher blastization indices. As for the Tatsen group, the results were very close and almost similar in the both groups, negative and positive. You have here a overall representation, graphic representation, an average done for all groups, negative, doubtful, and positive. And overall, the average shows that to each of the plant's extracts or mitogens, classical mitogens, the blastization indices were higher. Brucella ovis infection did not significantly increase the spontaneous blast transformation, either in positive, doubtful, or negative group. The highest responses were obtained for Taraxum officinale and Equisetum palustre. And even though the initial hypothesis was that serologically positive RAMs may have an enhanced reaction to immune-stimulating plants, due to this reactive status to the intercellular pathogen was invalidated. So even if the positive group showed higher blastization indices, they were not statistically supported. Thank you very much for your attention.
Yeah, okay, thank you.
Thank you very much.
Yes, nice presentations. Very fruitful and valuable information share us, and thank you. And
thank you.
Gülsüm Yaldız hocam burada mı acaba?
Yes.
And, tamam hocam. Ben mikrofonu size ve Marina hocaya bırakacağım da onun
I
için.
came back.
Okay,
Sorry
thank
for
you.
that. We linked from the same computer, not to overload the system. And now I don't know why.
Yes,
are you going to share the program, or shall I share the program on the screen, Sevgi, so we can?
Yes, now I am sharing.
So now we would kindly invite, I think, Mahmoud Samlitsa, sorry for the pronunciation, I don't know how it's pronounced, to present us the results obtained in agromorphological and yield properties of different fenugreek genotypes and cultivars under irrigated and dryland conditions.
Yes, there's chairperson and the other person. Welcome to my presentation.
Okay, he's not with us.
Can you hear me?
Yes, I can hear you.
Okay. I will present the topic about agromorphological and yield properties of different fenugreek genotypes and cultivars under irrigated and dryland conditions.
I just would kindly ask you to put the presentation on full screen, please.
Full screen, okay.
Thank you very much. And now the floor is yours to start the presentation. Thank you.
You are welcome. First of all, fenugreek is an annual crop and member of the family Fabaceae, the order Leguminaceae. Trigonella genus includes nearly 100 species, and out of which 50 species grown naturally in Turkey. The important fenugreek producing countries are India, Morocco, Egypt, Ethiopia, and Turkey in the world. Fenugreek leaves and seeds have been used extensively to prepare extract and powder for medicinal uses. This plant is primarily used as a spice in India and countries of the Mediterranean regions. Seeds of fenugreek contain tannic acid, fixed and vegetables oil, especially diosgenin, trigonellin, trichocumarin, trimethyl-coumarin, steroidal saponin, such as cytogenin, and trace of trigogenin and vitamin A. Some of the therapeutic uses of fenugreek include its use as anti-ulcer, wound healing, immunomodulatory, antioxidant, anti-diabetic, and anti-cancer. This study also was carried out to determine the morphologic and yield properties of 18 genotypes and 2 controls of fenugreek under irrigated and dryland conditions. We use 18 fenugreek genotypes and 2 cultivars in our study. Table B shows the genotypes, accession code, and country. Table 2 shows the average climatic data and soil table 3 shows the soil properties of experimental area. All the selected genotypes and cultivars seeds were sown on April 18th, 2019, and were planted on 30 centimeters at plant to plant and row distance of 10 centimeters according to randomized compliant block design with 4 meters long and 0.3 meter interplant spacing. As the base fertilizer, diammonium phosphate was added at 3 rate of 16 kilogram and total 40 kilogram hectare as fertilizer application was applied to place as top fertilizer during the soil preparation in 2 splits in April and of May. These genotypes were regularly irrigated and in irrigated conditions to demonstrate good progress in its period vegetation at regular interval of 3 to 6 days. Before harvest, the morphological and yield properties were measured, and these genotypes were harvested at physiological maturity from late of July to early August. The first harvest was made in PI 448 in irrigated condition and PI 264 genotype in dryland condition. The last harvest was made in PI 73 genotype in irrigated condition and PI 85 and PI 15 genotypes in dryland conditions. The result was subject to the analysis of variance, and least significant differences was used to find differences among the genotypes and controls. The statistic of jump 13 was used to determine statistical significance. The obtained result from fenugreek analysis and cultivars were in table 4 and 5 in irrigated and dryland conditions. Table 5 shows that morphological properties of fenugreek genotypes under irrigated conditions. This table shows the growing day, 50 growing days, first flowering day, and plant height, number of branch, first fruit linking, plant length, pod weight, and pine days, and harvest day. This is the morphological properties of fenugreek under dryland condition. Especially we explain the yield values because it is so important to fenugreek. One of them is biologic yield. It is changed from 191 to 713 kilogram in irrigated conditions. The highest biological yield was found from PI PI-15 and PI-76 genotypes under irrigated and dry land conditions, respectively. The lowest values were obtained from PI-38 and PI-33 genotypes under dry land conditions, respectively. Harvest index sank between 16 to 28 in irrigated conditions, and it was ranged from 19 to 32 in dryland conditions. Pot number varied from 8 to 20, and the highest value was found from PI-40 genotype in irrigated conditions. For dryland condition, pot number changed between 4 to 10, and the highest and lowest values were obtained from the same genotypes as PI-64 and PI-33 genotypes respectively. Seed number per pot sank between 10 to 16 number and between 10 to 15 number and under irrigated and dryland conditions, respectively. The highest seed number was found from PI-64 and the lowest value was obtained from PI-20 genotype for irrigated condition. The highest and lowest values were found in PI-49 and PI-38 genotypes under dry land conditions. Pot weight ranged from 0.19 and 0.33 gram and 0.21 and 0.39 gram. Seed weight per pot varied from 0.12 to 0.20 gram and 0.14 to 0.26 gram in irrigated and dryland conditions. Both pot weights of seed weight per pot was found in 50 cultivars as the highest values and was determined from PI-20 genotypes as the lowest value in its conditions. Seed yield is one of the most important factors in fenugreek, 37 to 142 kilogram in irrigated conditions and varied between 31 141 kilogram in dryland conditions. The highest values were obtained from PI-15 and PI-76 genotypes, and the lowest values were determined PI-020 and PI-38. 9 genotypes were found over the average of genotypes in irrigated conditions, and 6 genotypes and cultivars were found over the average of genotypes in dryland condition. In addition, 5 genotypes were determined over the average values of genotypes in 2 conditions. 1,000 seed weight was found between 18 gram and 11 to 21 gram among the fenugreek genotypes and cultivars in irrigated and dryland conditions, respectively. The highest value was found in PI-73 genotype under these 2 conditions. The lowest values was determined in different genotypes as PI-20 genotype under irrigated condition and PI-49 under dryland conditions. This table shows the yield values of fenugreek genotypes and cultivars. As a conclusion, we have observed considerable variability among fenugreek genotypes. They show different effect in irrigated and dryland conditions. The highest values were found from irrigated area in terms of pot number, seed yield, and biological yield, and 1,000 seed weight harvest index were found from dryland conditions. This study demonstrates that fenugreek genotypes and cultivars can be successfully grown as a culinary space under both ecological conditions. PI-15 and PI-76 and PI-73 genotypes produce good ground phenology and yield properties compared to the rest of genotypes. Among the important genotypes, especially PI-76 genotypes was the best efficient to irrigated and dryland conditions. These genotypes can be selected as superior line in the next breeding program and variety candidate. Thank you very much.
Thank you very much for your very interesting presentation. I think that we should ask now if anyone has any questions to you about your results.
No question. No question. Thank you.
Apparently no questions. Hello, Mihaela, how are you?
Hello, Professor. I'm fine.
I think then we should then pass the word to Gülsüm Yaldiz. She's going to present, or you're presenting the next paper entitled Variation in Some Quality Properties of Different Basal Genotypes.
Dear Professor, I will present this our presentation.
Okay, so we are listening then. If you would be so kind to put your presentation on full screen, please.
Oh, yes, okay. I think this is okay.
We can't see your screen yet.
Okay, thank you very much. The other presentation names is variation in some quality properties of different basal genotypes. Basal is the most common.
We cannot see your screen yet. Can you share with us so we can see your presentation, please?
Okay.
Now it's better and full screen. Okay. Thank you very much. Thank you. So we're listening to your basal results.
Thank you very much. Basal is an important medicine. Basal is the most common space in Lamiaceae family. Ocynum genus includes nearly 150 species, and within the space Ocynum basilicum, there are a number of varieties that differ in the general morphological structure and texture, and these varieties are cultivated in the world. Basil is an important medicinal and aromatic plant which is cultivated in many parts of the world. It is used for treatment of dry mouth and dental complaints, diarrhea, chronic dysentery, and respiratory disorders. It is also effective in the treatment of fungal disease and stomach discomfort and influent antitussive, diuretic, and expectorant roles in medicinal approach. In addition, ceasing nasal bleeding and preventing constipation, good for fatigue and insomnia, and uses for healing migraine, headaches, and incomplete paraplegia were reported. Many researchers reported that morphologic properties, yield, and yield components of basil change in terms of plant height, fresh herb yield, and its components under different ecological conditions. Several studies deal with grouping of basil varieties based on only one nutrition characteristic according to chemotype. The main objective of our work were to determine the essential oil, protein, and mineral content of different basil genotypes, to compare local cultivar with different origin basil genotypes in terms of these properties. We use 35 different genotypes, and for controls in this study, experiment was carried out according to augmented experiment design. It is include 5 blocks, of which every block had 7 genotypes and 4 controls. 6 of these genotypes shown 15 April 2018 years. Before transpiration of seedlings, 8 kg diammonium phosphate was applied to plots as a base fertilizer, and after transpiration, nitrogenous fertilizer as 6 kg ammonium nitrate was applied to plants in 2 splits in April and July. The first harvest was at the beginning of flowering and started from the first week of July until the 3rd week of August. We also determined the essential oil contents of these plants by using a Krevinger device. Protein analysis was conducted with Kendall method, and protein content was described by Brzezinski-Wirdek mineral content was determined as reported by Yaldız. This table shows the basal genotypes and cultivars information. The essential oil content of basal genotypes and cultivars changed from 0.10 to 1.06%. The highest essential oil content was from MS09 genotype, and the lowest essential oil content was found from PI00 genotype. Average of examined genotypes was 0.52%. 11 basal genotypes were determined higher than control means. Protein content changed between 9 to 18% among the basal genotypes and cultivars. The highest protein content was found from Moonlight and followed by Bolu genotype. The lowest protein content was observed in Midnight cultivar and followed by PI78 genotype originated from Turkey. Total average of genotypes was 13.02%. Cultivar average was 12 to 32%. 25 different genotypes were determined over the average of cultivars. Mineral contents, we determined the mineral contents of basal genotypes and cultivars as chlorine, phosphate, sulfate, sodium, ammonium, potassium, magnesium, and calcium. Chlorine content was ranged from 2060 to 10,900 milligram, and the highest chlor content was found from PI90 genotype, and the lowest chlor content was observed from PI78 genotype. Phosphate, the highest phosphate was found from Moonlight cultivar, and the lowest phosphate was found from MS84 genotype. Sulfate was found 320 and to 3,273 milligram, and sodium changed between 22 to 54 milligram among the basal genotypes and cultivars. The highest and lowest sodium values were found in the same genotype with chlorine content. Ammonium was found between 0.69 to 125 milligram. It was determined in 8 genotypes and 1 cultivar. The highest value was found from cultivar. Potassium content varied between 5,500 to 15,900 milligram in basal genotypes and cultivars. The highest values were found from PI12 and Midnight cultivar. The lowest values were obtained from PI84 and followed by Moonlight cultivar. Magnesium content ranged from 700 to 2,600 milligram. The highest and lowest values were found in Midnight and Moonlight cultivars respectively. Calcium content of basal genotypes and cultivars changed between 2,800, 11,700 milligram. The highest value was found in Midnight cultivar, and the lowest value was obtained from PI84 genotype. We looked at previous studies, and they are sometimes found similar with our result or different. This can be differences can be explained between our result and early studies, maybe due to the different environmental, genetic factors and applications. Conclusion: the highest essential oil and protein content were found in the different basal genotypes and cultivars. In this context, MS09 and PI91 genotypes were recommended for high essential oil more than 1%, and Moonlight cultivar and Bolu genotypes were suggested for protein content over 15%. In this study, Midnight cultivar had higher mineral contents as sulfate, ammonium, magnesium, and calcium among the basal genotypes and cultivars. On the contrary to moonlight cultivar had lower values in terms of chlorine, phosphate, sodium, and magnesium contents. It was also shown that sweet basal leaves are rather rich source of potassium, calcium, phosphate, magnesium, and chlorine, and potentially bioavailable for human consumption. Thank you very much for this presentation.
Thank you also for your presentation.
Thank you very much. I have a question, if I may. Are you going to continue, because you're showing us some indicators which differentiate different genotypes of plants? Are you going to proceed with obtaining some products for human use or medicinal use from these products based on their content?
Yes, we are going to studying some disease, especially Alzheimer's disease for human. We have a project with medicinal faculty, and we look something good result.
Thank you very much, and thank you for your presentation. I think that well, maybe Diana should introduce the next paper. Are you kind of coordinator with me here, or Sevgi, or who is?
Yes, I can.
I do not want to monopolize everything.
Yes.
If it is okay. Okay.
And the next paper is the same group?
Yeah, the same group, yeah.
I will present also this.
Okay, so we invite you. I must apologize. Could you pronounce your name for me? Mahmoud and?
Mahmut Çamlıca.
Çamlıca, okay. So,
Yes.
dear colleague, please tell us something about yield components, and some quality properties of fenugreek cultivar and genotypes. Do you agree with me, Mihaela?
Yes, of course, I agree.
Okay, so thank you very much. Yeah, the floor is yours. So just share your screen with us.
Okay, I will present
Full screen, please. Perfect.
Thank you. This presentation is yield components and some quality properties of fenugreek cultivar and genotypes. Especially, I will pass the introduction because the first presentation about the fenugreek. I will start directly materials and methods. Plant materials were obtained from Ankara University. We use 2 selection lines and 1 cultivar. Field experiment was carried out during 2013 and 2024 years according to split block design with 3 application at all the university experimental farms. Main plots were assigned to lines and cultivars, while side plots were devoted to sowing date. Base fats are the same as the first presentation. 6 kg diammonium phosphate and 2 kg calcium ammonium nitrate were used, and then 2 kg calcium ammonium nitrate was applied to top fertilizer before flowering date. We use also determined the crude protein content and crude oil rate as reported by Çamlıca and Yıldız. Statistical analysis was analyzed using JAMP JMP 13 statistical software, and results were compared with least significant differences tests. Also, correlation and PCA analysis were used to relationship between the fenugreek cultivar and lines for the examined parameters. Results, firstly, plant height changed between the 30 to 63 centimeters. The highest plant height was found in the plants sown on first sowing date in line 2 and followed by second sowing date in Güraslan cultivar. The shortest plant height was observed in the fifth sowing date in line 2 and followed by sowing date of line 2. Branch number was found between 1 to 4 among the fenugreek cultivar and lines. Pot number was checked between 3 to 11 numbers. The greatest pot number was observed in fenugreek cultivar of the second sowing date, and the lowest value was obtained in line 1 of the fifth sowing period. Seed number per pot, significant differences were found among the fenugreek cultivar and lines in seed number of per pot in different sowing days. The highest seed number was recorded by the plant sown on second sowing date in Gülistan cultivar and followed by first sowing date in line 2. The lowest seed numbers was seen in the fifth sowing date in line 2. Pot length sank between 14 centimeters and the shortest pot length was found 10 centimeters. Biological yield, there was found significant differences among the fenugreek cultivars and lines in terms of biological yield. The biological yield was found between 154 378 kilogram and mean was not as 249 kilogram. The highest biological yield was found from second sowing date in line 2 and the lowest value was observed in fifth sowing date in Gülistan cultivar. Harvest index sank between 19 to 43. Seed yield was ranged from 45 to 86 kilogram. The highest seed yield was observed from second sowing date of line 1 and followed by third sowing date of line 3 with 85.98 kilogram. The lowest seed yield was found from fifth sowing date of line 2 and followed by line 1 and Gülistan cultivar with 52 kilogram and 54 kilogram respectively. 1000 seed weight changed from 17 to 20 gram. The fourth sowing date of Gülistan cultivar had maximum 1000 seed weight with 20 gram and followed by 20 gram and 20.50 gram and 20.47 gram of third and second sowing days of Gülistan. Generally, 1000 seed weight of Gülistan cultivar was found higher than line 1 and line 2 in all sowing dates. First sowing date of line 2 give the lowest 1000 seed weight. Protein content was we didn't record any such a significant difference to protein content of fenugreek lines and cultivar. The highest protein contents were found from third sowing date of Gülistan cultivar and fifth sowing date of line 1 with 10%. The lowest values were obtained from fourth sowing time of Gülistan cultivar and third sowing date of line 2 with 9.64%. Crude oil rate, there was not found differences in terms of crude oil rate of fenugreek cultivar and lines among the different sowing dates. Crude oil rate ranged from 2.07 to 4.93%. The highest result was found from third sowing date from line 1 and the lowest value was observed from fifth sowing date of Gülistan cultivar. Correlation matrix and principal component analysis of fenugreek cultivars and lines. The correlation matrix were the relationship among the fenugreek cultivar and lines. Totally 5 significant relationships were found from examined properties as positively or negatively. The highest positive correlations were found between pot number and pot seed numbers, between branch number and 1000 seed weight, and a negative correlation was observed between branch number and seed weight. To hide the similar negative correlation were found between 1000 seed weight and seed weight, also found between biological yield and harvest index. Principal component analysis showed two principal components according to 100% of total variation of morphological and yield attributes in fenugreek cultivar and lines. PC1 showed 56.97% of the total variation and strongly influenced plant height, pod seed numbers, pod length, pod number, crude protein content, and crude oil content. Also, PC2 explained 43.03% of total variation, and it was strongly associated with branch number, 1000 seed weight, biological yield, and seed yield and harvest index. Fenugreek cultivar and lines evaluated with examined properties. Gyrastan cultivar was positively associated with branch number, pod length, and total 1000 seed weight. Line B was associated positively with harvest index, crude protein content, and crude oil content. And line 2 was positively associated with plant height, plant number, pod seed number, biological yield, and seed weight. Gyrastan cultivar and line 1 were associated with PC1, and line 2 was associated with PC2. Result of the PCA analysis, line 2 can be chosen as the best among the cultivar and lines in PC in terms of seed weight. Conclusion, fenugreek is sensitive crop to sowing date in different ecological conditions. The sowing date increased or decreased the fenugreek morphology and yield properties as significantly. To find the most suitable sowing days, studies can be conducted to growing place. Generally, it was found that second and third sowing days gave the highest morphological and yield properties, and this can be recombined. Seed yield had the highest values in line 2 except fifth sowing type, so line 2 should be evaluated by the farmers or breeders for different sowing time. Thank you very much.
Thank you very much. Any questions? No questions at this point. I would ask you something again. I don't want to bother you too much, but it was very interesting that you presented now some data on the harvesting period and yields of this plant, which is a spice, if I'm correct. Are you planning to write this down for the farmers to guide them in how they should proceed with their cultures? Like a brochure indicating what's the best for them to do? Help them get better yields of the crop?
Now we work about 18 genotypes and 3 cultivars on fenugreek to recommend farmers, I think.
Are you going to write down
Yes.
what they need to do to have better cultures, better yield?
No, next time we will have, I think.
Okay, thank you very much.
Thank you, you are welcome.
And now I think we should invite the next paper, which is a paper on garlic, Allium sativum essential oil chemical composition and its potential effect in food and medical applications. Is anyone from the group presenting this paper?
Actually, Professor Marina, Dr.
Yes.
Zakia should presented yesterday, but she was not available. So we changed the program.
Yes.
We write the name in this today. I think I write the message, but
Maybe
yes.
we should move to the next presentation.
Maybe,
and then
maybe
they will respond to the message afterwards.
Okay.
So, Diana? Would you like to introduce the next paper?
Yes, the next paper should be the chemical content of Salvia nemorosa growing around the Van Lake, and we will invite Professor Murat Tunçtürk if he is available to speak. We invite him to present the paper. Professor Murat, are you here?
Instead of Professor, I will make a presentation, Professor Veyde Tunçtürk, I may.
Okay, so I invite you to present.
I greet to the session chairman and all participants. I will make a presentation, name it.
Can you please share your screen? Yes.
Yes, okay.
And make it full.
Yes, okay.
Full
The
screen.
chemical contents of Salvia nemorosa growing around Van Lake. Salvia, one of the largest genera of the Lamiaceae family, contains about 900 species of herbaceous, shrub, perennial, rarely annual or biennial, which are often heavily aromatic and grown in warm and moderate regions of the world. Members of this genus in the Mediterranean, southeast of Asia and Central America are distributed. Many species of the genus Salvia are to be found in East Anatolia, Turkey. Nutritional value and mineral compositions of wild edible plants have been investigated for food security and human health. The main components of essential oil, beta-caryophyllene, germicrin B, caryophyllene oxide, cis-beta-farnesin, and germicrin D. Salvia is famous for its medicinal properties, and in traditional medicine is used to treat colds, wounds, and skin infections, headache, brain anemia, memory impairment, and hepatitis. Also, many species of salvia are used as herbal tea and as a flavoring for food, as well as in cosmetics, perfumery, and pharmaceuticals. Leaves of Salvia numerosa have been used in Turkish medicine to stop bleeding by applying externally. Aromatic plants and essential oils have been highly valued since prehistoric times of medicinal properties and food preserving power. More recently, aromatic plants and essential oils have been shown to be associated with a wide range of pharmacological effects such as spasmolytic, carminative, anti-inflammatory, hepatoprotective, antimicrobial, and antioxidant effects. In fact, aromatic plants and essential oils are increasingly used by the pharmaceutical, food, and cosmetic industries. Elements such as potassium, phosphorus, calcium, magnesium, and sodium are very necessary elements for human and animal health. However, some elements are essential for the organism. In their absence, both growth and reproduction are stopped. They have poison effect when they reach certain levels. The heavy metals such as chrome, lead, cadmium, cobalt, mercury, and nickel in the plants. In this study was aimed to determine the nutritional values and mineral compositions of Salvia nemorosa species which grow as wild plants in the one region of Eastern Anatolia, Turkey. The plants were collected from natural flora in 2013 and botanical identifications were made by the Department of Biology, Science Faculty, Van University, according to Flora of Turkey. Some information for Salvia nemorosa species are given in Table 1. Lamiaceae family, local name, Dlaga, Adaçayı, Kaşketenik. Benefits, wound healing, diuretic, stomach, used parts, livers, locality 2,315 meter. Electric muffle furnace set at 515 degrees centigrade were used for determination of total ash contents. Caldal apparatus and method were used to find the nitrogen contents of the samples. Crude fiber analysis were accomplished by AOAC method. Atomic absorption spectrometry was used for determination of mineral contents. Phosphorus was determined by molybdenate vanadate, and sulfur was observed according to the Michel method in conjugation with a UV visible spectrophotometer. In these, the nutrient contents of Salvia nemorosa that are important for human health, for the prevention and control of diseases were investigated. The total ash, total nitrogen, crude protein, pH and crude fiber contents of Salvia nemorosa situated in 1 flora were determined as 7.0, 0.183, 8.153, 5.16, and 4.40% respectively. Conclusions obtained from our study are compatible with the previous scientific reports on medicinal and aromatic plants. In this study, sodium, magnesium, potassium, calcium, phosphorus, and sulfur contents were determined as 0.84, 3.26, 15.20, 11.17, 2.14, and 0.48 gram per kilogram. The findings obtained from the research are partially similar to the results of the researcher. The daily sodium need per person is of 5 and 15 grams. It is known that hypertension is triggered by excessive consumption. The World Health Organization declared that the minimum daily potassium requirement is 3.51 gram per kilogram of a person for the protection against heart disease and the regulations of blood pressure in adult daily magnesium consumptions, but its amount must be up to 300 and 420 milligrams. In the present study, we also determined some micronutrients element contents as seen in table 4, the concentrations of manganese, iron, copper, and zinc. In analyzed samples were determined as 15.5.46, 322.72, 28.53 and 15.73 milligram per kilogram. In this study, determined concentrations of nutrients are in accordance with previous studies. Copper is necessary for proteins and enzymes as a cofactor for plants to grow up, but its excess intake might cause health problems. 2.5 milligram of daily copper intake can meet the daily requirement of adults. Zinc is required for normal growth, a healthy immune system function, DNA synthesis, new protein synthesis, and cell division, as well for different chemical reactions. In the present study, we also determined some micronutrients element contents as seen in table 5. the concentrations of chrome, cobalt, cadmium, and lead in analyzed samples were determined as 1.14, 0.19, 0.11, and 0.12 milligram per kilogram. Our findings with findings of previous studies are within the same limits. These metals, known as heavy metals, and their hazardous effects on living organisms in certain quantities were reported. Doğan determined that accumulation of 6.15 milligram per kilogram cadmium in onion plant. Also, researcher did determine that this situation can cause significant health hazards. In addition, it is stated that more than 1.0 ppm cadmium is toxic in plants and it has poison effects on people who regularly consume plants containing more than 3 ppm cadmium. In order for our body to function properly, there are certain amounts of macro and micronutrients that we must take from the foods we eat. But these nutrients must be within the tolerance limits determined by World Health Organization. Otherwise, it might have adverse and undesirable effects on human health. When heavy metals exceed the concentrations limit, they act toxic. The findings showed that Salvia nemorosa plant was found to be within tolerance limits for heavy metals. Furthermore, it is important to consciously make of plant consumptions by considering that heavy metals deposited in excess on humans and animals may be toxic. Thank you for attention.
Thank you also, Professor Veyde Tunçurk, for your presentation. If anyone has questions.
Yes, please.
Yes, sorry, Diana. I want to make an announcement to all our participants who are in Salon-B. Now, when we close this session, all the participants should be online in Salon-A, and there will be a poster session and closing speech in Salon-B. Now they finished the session in Salon-A. They are waiting to finish our meeting in Salon-B. I think we are the last presenter.
So if I may, I would like to invite
yes,
Hasna.
I am sharing now, Professor Marina.
Okay.
Yes, I think the last one.
Yes, it's the last one.
Please. So Hasna, you can present your paper.
Please, I am Zakia Bubshisha.
Okay.
Oui.
Yeah, Professor Zakia, you can present your paper.
Thank you.
Okay.
But sorry, Dr. Zakia, we have only 10 minutes. Please just conclude your research for us. Okay. Could you please share your screen, Dr. Zakia?
Recently, I will share and sample of system, and so for 2 days I can go only to home and sleep because it's too difficult to manage.
Yes, okay.
Online. The first place is really easy.
It's okay.
Yeah, please make just full screen. Could you please? There will be a button in the right side.
I can commence.
less effectiveness.
Okay.
And we couldn't manage
Yes.
Yes, please.
Okay.
Thank you. Good morning, all. My present MVT talk is on garlic essential oil, chemical composition, and its potential effect in food and medical application. This presentation is made under the direction of Jama from the Lille School of Chemistry in France and Professor Halel from Polytechnic School of Algiers. Essential oils have been used across many civilizations for a variety of purposes, not only in the manufacture of perfume and cosmetic, but also for medical purpose to heal body and soul. At present, about 150 essential oils are commonly marketed compared to 350 years ago. In recent years, global production of essential oils has increased significantly. Recently, it has estimated at over 350,000 tons. The major producing countries of essential were Brazil, followed India and China. The most important essential oils in tons were orange, mint, lemon, eucalyptus. The world's largest essential oil by tonnage is orange essential. It is produced in excess of 50,000 mainly from Brazil and Florida. Next is mint and followed by lemon. The essential, the major destination of essential oils were the food market, the perfume and cosmetic market, and the aromatherapy market. For the food market, the essential is the beverage and distillation, and the most commonly used is mints and citrus. Concerning the perfume and cosmetic market, the use of essential oils is low compared to synthetic products, but there is a special case for organic cosmetics, which the consumption more is the 3% with an and the growth of 8%. Concerning the aromatherapy market, 2% of the world trade in essential oil, but the aroma market is growing. In this presentation, we interested for garlic oil. Garlic oil
Professor Zakia, if it is okay, can you please just make your conclusion of the paper, because we have one more presentation and we need to manage the time, please.
Yes.
Thank you.
About this presentation, the garlic oil was used in different applications in food science, such as flowering properties and preservative and food additive. Also in application in medical science, such as antimicrobial, antioxidant, antitumoral, antithrombotic, antidiabetic. Thank you for your attention.
Thank you for your presentation. Now we will move to the last presentation, and I would like to invite Hasna to present her paper analyzing of bioactive compounds of cultivated garlic, Allium sativa, and onion, Allium cepa in Algeria. Please, Hasna, you can present your paper.
Thank you.
Please make your presentation, share your presentation. And make it full screen. It is possible.
Okay. Firstly, I would like to thank Professor Nazim and Dr. Sevgi for this organization. So I will present a part of my PhD, which is analysis of bioactive compound of cultivated garlic and onion in Algeria. So Algeria, by its biographical...
Hasna, beni duyuyor musun? Hasna.
What?
Hasna, can you hear me?
Yes.
Please directly go to conclusion. Directly go to conclusion.
Okay.
So people waiting, I am so sorry. Some people waiting another salon. We should start the closing ceremony. After here, you will enter again the Salon-A. You can find the ID and password from the website. Yes, directly conclusion. I am so sorry.
So as conclusion, during the phytochemical screening, there were very significant difference, both qualitative and especially quantitative, between this spice. Onion was rich in polyphenol, flavonoid, tannin, and possessed the higher antioxidant activity. The good antioxidant activity for onion in comparison with garlic could be related to the higher content of quercetyl derivative. And thank you. I would like to sincerely thank the staff of Pharmaceutical Biochemistry and Clinical Laboratory for the University of Medicine and Pharmacy of Romania, especially Professor Marina Spinu and Mihaela Nicula for Department of Infection Disease from Cluj-Napoca, Romania, for providing necessary facilities of characterization analysis for HPLC. And I would like to thank Professor Nazim Şekeroğlu for everything. Thank you.
We thank you also, Hasna, for your words.
Thank you. Thank you.
Please, I have a question, please.
Please connect to Salon-A, please.
Okay, so, okay.
You can find the password and the meeting ID from your emails and website.
Please, I have a question. Please. Should I leave the Salon-B to join the Salon-A for poster presentation?
Yes, now we go all in Salon-A, so we will leave these presentations.
Yes, thank you.
You're welcome.
Şimdi, prezentasyon.